Thank you, everybody, for coming, and thanks again to the organizers, the invitation to come to this exciting city, Beijing. As Dr. Wang mentioned, I'm going to talk to you about AAA, or abdominal aortic aneurysms, and I have the good fortune to be involved with both genetic epidemiology studies as well as basic science studies. So why do I show you this picture on the left of this famous both guitarist as well as physicist? Well, unfortunately, he's also one of the most famous people to have succumbed from an abdominal aortic aneurysm. So what is it? What is a AAA? It's a weakening of the main vessel in our body, the aorta. Most often happens in the infrarenal, right below the kidneys, where you get a dilatation or a weakening of the blood vessel that continues to grow over time. And the worst case scenarios is that it ruptures. If it ruptures, 80% of the people never make it to the hospital because they die instantaneously from bleeding. So it is relatively common. It happens in 5% of men over 60 years of age and about half as many women. And it is associated with many of the same risk factors that we associate with atherosclerotic disease. There's a history within the families, so arguably potentially a genetic component to it. I told you it's associated with age. It most often will be seen in the 5th decade of life and then beyond. And then, of course, it has male dominance. And then, importantly, the most modifiable risk factor associated with AAA is actually smoking, cigarette smoking. It is 90% of the individuals who present with a AAA have used tobacco at some point in time in their lives. So it is the most associated disease with smoking other than lung cancer. And then most, maybe interestingly, is the fact that diabetes is actually a negative risk factor for AAA. So there's something distinctly different about AAA than regular garden variety atherosclerosis. Human pathological studies, as well as animal models, have taught us a lot about the pathophysiology of AAA. It includes transmural inflammation, so it's an inflammatory disease. It includes loss of the extracellular matrix, but also, importantly, the smooth muscle cells. You lose smooth muscle cells, and therefore you have a weakening of the structure of the blood vessel at that component. And then that leads to a progressive dilatation. If you think about it, it's like the bubble in your bicycle tire. Once it starts, it just continues to grow due to the pressure inside the blood vessel. And again, the most unfortunate situation is that it ruptures. We don't know enough about the molecular mechanisms of AAA such that we don't have a medicinal therapy for AAA yet. The only therapy for AAA is actually surgical repair. And so therefore, when, as I said, the disease continues to grow and it reaches a point where the risk of a surgical complication is outweighed by the risk of rupture. And so therefore, it's actually managed, if you find it, it's managed by just serial imaging studies. And again, once it reaches a certain size, then you are recommended for surgery. Otherwise, we have no therapies for AAA. So I'm going to walk you through a couple genetic studies, but also some molecular biology studies to hopefully help us understand not only pathophysiology, but potentially also to identify some novel therapies going forward. So it was in 1977 that Martin Clifton first described a family that he was taking care of, 3 brothers who presented with a AAA. And he was the first to actually, in 1977, to say, maybe there's a genetic component to AAA disease. And since then, there have been many family studies as well as twin studies to say that there's a large genetic component. Up to 70% of the heritability of AAA is actually genetic, such that your first degree relative of a AAA has a 2 times risk of developing AAA itself. So that gives me an opportunity to introduce you to something called the Million Veterans Program. So I, in addition to working at Stanford, I work for the VA hospital. VA, the Veterans Administration hospitals, are the closest thing we have to socialized medicine in the United States. It's over 170 hospitals across the United States that have been developed, a system developed to take care of our veterans of the military. And so at any given time, roughly 9 to 10 million people take care within the VA system. There's also a research component to the VA, such that there's a lot of research studies from basic science all the way through epidemiology studies. And it is within the VA that we decided to develop a cohort, a genetic cohort, that could be tied to their health record and therefore potentially be a biobank for all kinds of studies, including cardiovascular studies such as AAA. So the goal was to recruit at least 1 million individuals to donate a blood sample so that we could use that for genetic material. And then again, importantly, across the United States, the VA uses the same electronic health record. So therefore, we could tie that blood sample to their electronic health record. And the other important thing is that because veterans receive not only good quality care within the VA, but they also oftentimes receive discounted care. And so therefore, they stay within the VA system for many, many years, if not their entire lives. So we have a longitudinal record of their health as well as their disease, thereby giving it a good opportunity for whatever disease we may be interested in, whether it's cardiovascular, mental health, or cardiorenal, or anything like that. So that's the goal of the Million Veterans Project, is to actually produce a information library where investigators can come and potentially use that resource to study whatever disease they're interested in. And as of last year, we actually recruited our 1 millionth participant, and we continue to recruit, where we have about 1,050,000 individuals at the current time. So with that, then, we have committed to genotyping everybody. And within our first genotype release of 250,000 people, we already could see that we had over 7,000 cases of AAA, the largest cohort of AAA ever assembled for a genetic study. And so we performed a genome-wide association study to look across the genome to see if there were specific spots or specific loci that are associated with disease versus not. And sure enough, we discovered 14 novel loci, so we more than tripled the number of loci that have been known to associated with abdominal aortic aneurysm. And I color-coded them for blue for extracellular matrix, green for smooth muscle cell biology, red for inflammation. And you'll notice that quite a few of those genes are associated with yellow, which means lipids. So surprisingly, we found a lot of lipid genes that might be involved with AAA. But not only, again, assembling this cohort could discover a lot of individuals with AAA, but it also put us at the table so we could talk to other cohorts around the world. And we assembled a large group of studies that have ever studied AAA, probably the largest number of AAA patients, such that we had over not only 19,000 within the Million Veterans Program by now, because we released more genotyping, but then assembled with everybody else, we had almost 40,000 cases of AAA compared to about 1 million controls. And as such, then, not surprisingly, we discovered many more loci associated with AAA. We quadrupled the number of loci. And probably most interesting, at least from a translational standpoint, one of those loci in both of our studies was associated with the gene PCSK9. And it was, and actually was a protein quantitative trait loci, which means that this genetic variation is associated with different levels of PCSK9 in the plasma, such that lower levels of PCSK9 seem to protect from AAA. So you would argue then, therefore, that could be potential therapeutic intervention for AAA. So therefore, we could then automatically go into the basic science lab and try and see whether PCSK9 is actually playing a role in AAA that had never been described before. So here's one of the models that we use. This is the porcine pancreatic elastase model. This is a model where you isolate the abdominal aorta in a mouse, and then you actually infuse it with an elastase, which actually causes a local inflammation and reduces the elastin layers. And you can monitor this over time, and you can see then it develops a AAA. And the pathologists really like this model because it actually mimics a human AAA very well. It grows over time. We watched this model over 4 weeks, and in blue you can see that it actually, sorry, the slides are advancing on their own. In blue, you can see that's the diameter of the aorta, which grows over time. And in the bottom, you can see from the left a healthy aorta, where you have in red the elastin layers, and in green the smooth muscle cell layers. And over time, the elastin layer is compromised. You lose smooth muscle cells, and all the way on the right, you see then the resultant AAA or aneurysm lesion. Well, the other reason we really like this model is you can actually layer on different risk factors. In this case, we're layering on smoking, and what this little video shows you is that these are the chambers that we put our mice in for an hour a day over the course of the study. And what you see is some smoke, or in this case, e-cigarette vapor that they're inhaling. And so they do this for an hour a day. And what I want to tell you is that this is associated with an increase in AAA function. In other words, it actually makes the disease worse, very much like the human situation. And just like the human situation, I told you AAA is associated with age. So if you take a middle-aged mouse compared to a young mouse and expose it to the same insult, you'll get a doubling of the size of the AAA. If you take a female mouse compared to a male mouse, it develops a AAA, but it's actually smaller, much like the human situation compared to the male mouse. And if you made an animal diabetic, the same thing with the human situation. For some reason, it is protected from the eventual development of that disease. So what we hope is that we can use this model, as well as all of its variations, to try and delve into the blood vessel wall and try and understand the molecular mechanisms that are really controlling not only the initiation, but also the progression of the disease. So with that then, I told you that in our genetic studies, PCSK9 came up as an potentially important factor. So we started to look in the blood vessel wall, in the actual aorta during the development of disease, what is going on. And not only do we see highly inflammatory gene expression here, IL-1 beta as well as LOX1, but we also saw increased expression of PCSK9 itself in the blood vessel wall. So there have been descriptions of vascular expression of PCSK9. Many of you know that most of our PCSK9 that's circulating in the plasma is produced by the liver, but other tissues, including the aorta, can make PCSK9. And the result of that lesion is actually increased PCSK9 in the plasma as well. I can't tell you today whether that is due to PCSK9 release from the aorta, or is it actually an inflammatory response, like an acute phase response produced by the liver as well. In the future, I hope to tell you more, but just to say that PCSK9 is turned on in AAA in the aortic wall, as well as elevated levels in the plasma. And so that begged the question is if we could somehow control PCSK9, in this case with a knockout animal, then could we actually modulate the development of the disease? And sure enough, in the PCSK9 knockout animals, we could greatly reduce the development as well as the expansion of the AAA lesion. That knockout is associated with an increase in smooth muscle contractile phenotype. 3 genes, ACT2, taglin, as well as myosin heavy chain, are greater expressed in the knockout animals within the actual lesion than the control animals. So you have more of that preservation of that contractile phenotype, as well as significant reduction in the inflammatory response, decreased IL-1 beta, IL-6, TNF-alpha, as well as LOX1, all within the actual lesion itself. Finally, we've started to look at single cell expression, again, in the aorta itself. And not only do we see less inflammation, less inflammatory cells, but if you actually looked at those macrophages in the bottom right-hand corner, you can see that there are different phenotypes. We have less expression of Ly6A type of macrophages within the actual PCSK9 knockout animals compared to the control animals, decreased inflammation like IL-1 beta, and then interestingly enough, you have more of the reparative type of macrophages compared to the inflammatory types, as shown by MSR1 as well as ARG1 within the actual lesion. So in conclusion then, I hope I convinced you that AAA is both a common as well as lethal disease. And since we don't have any medical therapies to date, there are several unmet needs. And that genetic studies as well as molecular biology studies can help and work in a complementary fashion to help us try and not only understand the course of disease, but also potentially identify novel targets for therapy. Genomic biobanks like MVP are going to be important as we go forward to help us understand a whole host of common as well as rare diseases. And that transcriptional profiling reveals the pathways that could be important in not only the initiation of disease, but importantly, the progression of AAA as well. And that finally, we would argue right now that PCSK9 could be a viable therapeutic target for AAA disease. The other thing, again, I want to say is that both of these types of approaches, molecular biology as well as genetic epidemiology, are both team sports. You can't do it on your own. You can't do it with a small lab. You need to collaborate with many types of individuals. And what I would say to all of you, especially the trainees, is that you would concentrate on the studies that you're doing, but open your eyes and open your ears to different types of studies, be able to read the literature, to understand what those other studies are telling us, and then incorporate that into your own work. And finally, I just want to thank just a small portion of my collaborators as well as the funding agencies that have helped us fund these studies. So thank you very much, and I'm happy to answer any questions.